This is Saccharomyces cerevisiae. Don't let the name fool you, this isn't a novel organism. Rather, it is the ever-common fungus, baker's yeast. Belonging to the family Saccharomycetiaceae, or budding yeasts, S. cerevisiae is the most meticulously researched eukaryotic model organism and, through the efforts of an international consortium of scientists, has had its entire genome completely sequenced since 1996. At the time, it was the first and largest complete genome sequence of any eukaryote, coming in at a massive 12,068 kilobases, or over 12 million base pairs packaged into 16 chromosomes. Presently, we employ S. cerevisiae in a wide variety of biotechnological and biopharmaceutical pursuits, due to it being easy to use, able to produce heterologous proteins, and amenable to genetic manipulation. It is used in beverage fermentation, bread making, and bioethanol production, and, most notably, it is used in the production of hormones like insulin, vaccines, and virus-like nanoparticles for the research and treatment of human papillomavirus (HPV). But where did S. cerevisiae come from? How has the yeast which has become so vitally important in the agricultural and biomedical fields come into evolution? The answer lies in a phenomenon known as whole genome duplication. Gene duplication is a powerful mechanism driving evolution. By duplicating genes, a redundant Lucis copy is created and is effectively ignored by natural selection, allowing it to accumulate mutations that are considered forbidden and be reborn with innovative function. The consequence of this, however, is polyploidy, or possessing more than two complete sets of chromosomes, which induces genetic instability that persists until mutation, gene loss, or genomic rearrangement returns the genome to normal. In the S. cerevisiae genome, analysis reveals the existence of ancestral duplication blocks, large, paired chromosomal regions comprised of chronological paralogs with unique interspersed genes. This is consistent with the model of whole genome duplication, which suggests that S. cerevisiae is a degenerate tetraploid which arose from duplication of 8 ancestral chromosomes and returned to normal function through small deletions of approximately 90% of its duplicated genome. This hypothesis is challenged by an alternative model, which suggests that these duplicated segments arose from numerous local gene duplication and deletion events. However, this theory has since been disproven. Scientists Kellis, Beeren, and Lander deduce that the best way to prove the existence of an ancestral whole genome duplication event in S. cerevisiae would be to find a similar yeast, let's call Y, that descended from a common ancestor in the same lineage, but which had diverged before the duplication event. This yeast would hence be related to S. cerevisiae by a 1:2 chromosomal mapping. To confirm this, 3 criteria had to be satisfied. 1. Almost all regions in Y correspond to 2 sister regions in S. cerevisiae. 2. The 2 sister regions contain chronological genes in the corresponding Y regions, and 3. Almost all regions in S. cerevisiae correspond to one Y region, and thus pair to a sister S. cerevisiae region. Cloveromyces waltii, a close relative that diverged before the duplication, was chosen as a model for comparison. Multicellular genomes are large, and hence are difficult to assemble into a single, readable sequence. Whole genome shotgun sequencing solves this by randomly breaking DNA into small fragments, with overlapping sequence reads reassembled by computer into contigs and ultimately entire chromosomes. This technique was used to collate K. waltii sequence information into 8 chromosomes, totaling 10.7 megabase pairs, for analysis. First, Kellis and colleagues compared both species' genomes to identify orthologous genes in synthetic regions, blocks containing matching chronological genes across both species. This was quantified by an algorithm, which divided K. waltii into doubly conserved synteny, or DCS blocks, which defined clustered regions mapped 1:2 with S. cerevisiae. This duplicate mapping was used to define sister regions in S. cerevisiae and identified 253 DCS blocks, tiling 85% of each of K. waltii's 8 chromosomes. Within a typical block, 90% of genes from both S. cerevisiae regions were found to have matches within the single corresponding K. waltii region, and were interleaved in preserved gene order and orientation, consistent with patterns of whole genome duplication after massive gene loss. Furthermore, these blocks were used to define 145 sister regions in S. cerevisiae that preserved ancestral gene order, spanning 88% of the genome and containing 457 duplicated gene pairs. From these paralogs, we can also observe patterns of functional change during S. cerevisiae's evolution, such as accelerated evolution of new, derived functions from old ones, decelerated evolution of genes that retained ancestral function, and unusual codon adaptations. Kellis and colleagues' findings prove the occurrence of an ancestral whole genome duplication event in the Saccharomyces lineage, doubling its number of chromosomes and returning it to normal ploidy through subsequent gene loss events. Whilst the window of opportunity for exploration by gene duplicates is narrow, stochastic gene evolution plays a fundamental role in the passive origin of new species, such as the Cereviceae we see today. Analysis of whole genome duplication provides the opportunity to study the long-term fate of a genome after duplication and shed light on its consequences, both positive and negative. It is important that we search for signatures of such an event in other organisms, including ourselves as humans, so that we may better understand its role in early evolution and where we are headed as a species.